Good morning. My name is Sergio Pena. I'm a professor of UFMG in the Department of Biochemistry. And I was called to introduce our speaker this morning since he works a lot with biological control systems. And actually, it's interesting that in 1966, I came to the Faculty of Medicine of UFMG, and I fell in love with the library. I'm sort of a library rat, you know, I've been all my life. And in the shelf there was an interesting book by a guy called Norbert Wiener. And this book was called Cybernetics. And I didn't know enough to understand everything that was in the book, but I knew that it was something very important. And in 1948, Wiener had defined cybernetics as the scientific study of control and communication in the animal and the machine. And for those who don't know, cybernetics is a word that comes from the Greek, which means government, which means that it's the government of the system. And since then, cybernetics has evolved in a biological sense, in a Darwinian sense, getting more and more sophisticated, and connects the fields of control systems, electrical network theory, mechanical engineering, logic modeling, evolutionary biology, neuroscience, anthropology, and psychology. And today it has advanced even further. And these days, Ray Kurzweil talk about singularity as the moment in which the machine and the man will merge into a new organism. I don't know if it's something new or if it's the demise of humanity as we understand it. Anyway, we're going to be hearing a lot about these things, and we're going to be hearing from the right person to do it, who is Professor Babatunde Ogunnaike, who was born in Nigeria, did his undergraduate studies in Nigeria, and then he did his master's and PhD degree at the University of Wisconsin, and went back to Nigeria and was a professor in Lagos for until 1988. And then he worked with DuPont in Delaware and joined the faculty at the University of Delaware in 2002 and became the dean of the College of Engineering in July 2011. He's a well-known personality. He was visiting professor at the University of Wisconsin again, and at the African University of Science and Technology in Abuja. He was associate editor of the IEEE Transactions and Control Systems Technology. He's also associate editor of the American Chemical Society's Industrial and Engineering Chemistry. His research focuses on modeling and control of industrial processes, the application of process analytical technology for control of pharmaceutical processes, identification and control of nonlinear systems, the interaction of process design and process operability, applied statistics, biological control systems, that's a lot, and systems biology with application to neuronal responses, and cancer on top of everything else. So now, Professor.
Thank you very much. Is this microphone working? It is. Okay. I'll hand this back to you. Good morning. Maybe I should say bon dia. I would like to start by thanking the organizers of this conference, Professor Pena, for inviting me to come. And as I look down the audience, I see old friends, and I've been able to put faces to names that I used to just send papers to have them review for me. Thank you very much that I can identify people here. Over the next 45 or 50 minutes, I'm going to be telling you a story. And the way things are, I can't tell you everything, not in the short period of time that we have, but I hope that I can at least get a few of these things across, and then we can have some time for discussion. I'm going to present my talk in 3 parts. In the first part is where I'm going to introduce the concept of homeostasis and how homeostasis is actually achieved using physiological control system. The theme of this part is that human life as we know it will not be possible were it not for control. Okay? Human life as we know it will not be possible were it not for control. That's the first part. The second part, I would actually try and go a step further and say that if feedback control is involved in something and you don't know it, if feedback control is involved and you don't know it, or you know it but you don't pay attention to it, you are likely to make a mistake if all you look at is input and output data. Okay? Most of you are control experts. In fact, everybody sitting in this room is a control expert. You will understand what I'm saying. So at that point, I will talk about pathologies and why a control engineering perspective is a very helpful perspective to take in studying pathologies and in providing means of doing treatment. Finally, the last part, I will then show you actually one example of what happens when a disease occurs as a result of a failure of a control system, and how control, as you and I know it, physical control, can actually help in restoring people to good health. All right, so then I will conclude, and then we'll have some time for questions. So I'm going to start with this introduction. And I want to start with a very interesting historical experiment. In 1775, there was a person named Charles Blagden who lived from 1748 to 1820, and some research associates, a dog and a beefsteak. And here's the experiment. They put all of these folks, the associates, like graduate students, a dog and a beefsteak, the kinds of things we do to grad students, right? All of them spent 45 minutes in a chamber that was heated up to 127 Fahrenheit. That's very hot. Guess what happened? The dog panted, right? Men sweated. All of them survived, but the beefsteak cooked. Okay? The implication was that living organisms adjust to whatever conditions they're in. It was very hot. The human started to sweat, and because of evaporative cooling, was able to keep the temperature down enough until the insult was removed. Beefsteak cooked. Okay? And so they coined the concept of homeostasis. Okay? Humans could do that, living things could do that. The condition of stable dynamic constancy. If I take a bowl, and I take a marble, and I move that marble off, and I leave it, it would come back and settle to that point because of the way it was designed. Think about that in a high-dimensional space. That's exactly what happens with homeostasis. You perturb it, and then all kinds of things work together to bring it back as close as possible. And how is this possible? If you, and I'm not going to spend too much time on this, but if you decompose all of us down to our elements, you're going to find carbon, hydrogen, oxygen, nitrogen, and phosphorus. That's it. It's a little humbling, isn't it? that in the end, that's really all we are. And then you can assemble all of that together to get the DNA. And you put all of that together at the cellular level, you have cells like this. You stack cells together, you get tissues. You stack tissues together, you get organs, and then systems, and then the entire organism level. I'm putting all of this here because as engineers, we understand hierarchies. chemical process control people understand the whole concept of an entire plant, and then units, and then subunits, and things like that. It's exactly the same thing in the human physiology. And so, why do we need control in physiological systems? Well, if the whole objective is to maintain physiological function, to keep your physiological variables within specs, you don't have the luxury of designing the system and leaving it there to survive on its own, because we are constantly being bombarded by all kinds of perturbations. If we could just show you the amount of pathogens in this room alone, most people would be afraid of breathing if you were able to see. And yet, none of us is killing over and dying right now. Why? Because we have a fantastic immune system. And all kinds of things can happen, and yet you'll be able to adapt to it. Occasional insults, persistent perturbations, the potential for departure from our limits is limitless. And so you have illness is your short-term departure, but still within your non-lethal limits. Chronic illness is when you have this long-term departure, but you're still within non-lethal limits. Many of you probably know people with diabetes, that the blood glucose is very high. It's not normal, but they're living with it. Or people with high blood pressure and things like that. Well, death occurs when you then completely get out of whack. So, for example, I'm a chemical engineer. I work in the chemical industry, and we all know that in producing any product, all your process variables have to remain within certain spec limits. Otherwise, your product is not going to be good. Well, the same thing happens in the human body. So, for example, with glucose, people adapt this. You have a certain target, and then if you're within this range, you're normal. If you're below, you have hypoglycemia. If you're above, you're hyperglycemia. And anywhere outside of this range at a sustained pace, then you have trouble. How do the physiological systems maintain their function? Well, they maintain them by the organization of what I'm about to present to you. I teach a course in biological control systems, and in fact, I remember in the 2008 IFAC Congress, I taught a short course, and you were part of that short course. I organize the physiological systems into 10, the way I teach it to students, and I organize them as follows. I have the master regulators, where the nervous system and the endocrine system. We don't have the time to go through all of these details, but it will be amazing to see how fantastic these systems are in doing what they do. They're the master regulators because they're the ones that actually orchestrate everything. They're like the conductors that conduct everything. This one uses electrical systems. This one uses hormones. The defenders and the protectors, the immune and lymphatic system, the integumentary system, that's your skin, and the musculoskeletal system. And then what I call the utilities that supply you with things you need and take away the waste and things like that. So you have your cardiovascular system, which is pumping blood through the system, the respiratory system, how I'm breathing, I take in oxygen and I release carbon dioxide. The digestive system, all of us had breakfast this morning, and you get fuel and energy for all of the things that you need to do, but then you develop waste and you pass the waste out, and the urinary system, and then the species propagator. I hate to put it to you, ladies and gentlemen, but by the time we cross 40, after we have lived long enough to pass on our genes to the next generation, it's downhill from then on. And we live long enough to pass on our genes, and then we sort of kind of fade away, and then the next generation does the same thing, and so on and so forth. That's the reproductive system. That's how that works. So I'm just going to show you very quickly how all of these guys contribute to the life processes, and you can take a look at all of these things, and I'll leave my slides behind if you're interested in looking at this a little bit more. So you look at the nervous system. Yes, it does overall control and regulation, but it's also responsible for memory and behavior. The endocrine system does chemical regulation, but for growth and development. Defense and protection, but also fluid balance and fat absorption. The point is, you have your main responsibility, but you also have other things that you do so that you have this redundancy, so that if you actually do something, something else can take over for you. But these are the kinds of things that go on in the human body. And again, just from that diagram, it is next to impossible for human life to occur, to be sustained without control systems. And all of those control systems have what you and I would recognize as control characteristics, which is what I want to show you now. Block diagrammatic representation. Every control system, as we know, has a sensor. In this room right now, we are maintaining the temperature, I would guess, at about 19 degrees Celsius. So somebody set the set point as 19 degrees Celsius. There is a sensor in this room that is sensing what the temperature is, and then it's sending that information to the controller, and the controller is sending the information to the heat, ventilation, air conditioning system that is then pumping cool air into this place. Same thing in biological systems. There are sensors, a controller, actuator, and control processes. And believe it or not, you would see configurations like feedback. It's all over the place. All over the place in biological systems. Feed-forward control, that's also there. Cascade control is very prevalent with hormonal systems. And I'm going to show you. And the physics of the way hormones are used in the body actually dictates that it should be cascade control. I'll show you an example when we get there. And then there are really unconventional multi-loop systems. These are not anything that you will see in a classic control textbook, but they're there because of the nature of the work that is being done. So here's a classic example of the block diagram that you will see, your control system. There's some kind of a receptor that does the measurement. There's a control center, and then there's an effector. So and you can see that at the organism level, organ level, and cellular level. So for example, some of the earliest work we did was with a neurophysiologist who was studying the blood pressure control system, and it's relatively easy to map this. The sensors are called the bioreceptor neurons. They are sensitive to pressure. They're stretch-sensitive neurons on your blood vessels, and their characteristics. This is not a cartoon. This is real. The characteristics of the firing of these neurons on the basis of the arterial blood pressure is represented by this sigmoidal curve. And then at the cardiovascular center, actually the nucleus tractus solitarius is what is called, the NTS, is where that information is then converted to parasympathetic activity and sympathetic activity that go to different aspects. I'm not going to talk too much about this. So you can see the sensor, the controller, the actuators, not just one, several actuators, and your control process. Many control engineers will recognize that diagram. Why is feedback so prevalent in biological systems? It is so prevalent in biological systems because, as far as I know, feedback is the only way for achieving disturbance rejection and set point tracking without knowing precisely what is going on in your process. So if you linearize, many of you will recognize this closed loop transfer function, and you can see that in the limit, as your G sub C tends to infinity, if you crank it up your high gain amplifier, this goes to 1, this goes to 0. So without necessarily knowing anything, you can use your feedback to get you to where you need to get to, and that's why it's the most common configuration by far. But of course, everybody knows the problem with feedback. When I teach my students about feedback control, I say, imagine driving down a busy street, main street on campus, when students are crossing in between classes and all of that. Imagine if you were driving using feedback. Oops, I just killed a pedestrian. Oh, and then you get the feedback and say, don't do that again, okay? No, you can't do that. That's the problem with feedback, because you only respond after the fact. And because of that, there are all kinds of things. Your control system is susceptible to all kinds of things. And then, if you have long delays, I use this as an example again to my students. So I know I'm spending a little bit of time here, but I think it's more important to get all of these things clear and then I'll speed up a little bit as we go. You enter the shower in the morning, and you turn the water on, and it's a little too cold. And then you add some hot water. But for the sake of argument, let's say it takes 5 seconds for the hot water to travel through to get to you, right? So, but then you're going to be taking control action every second. So you turn it, it's supposed to be enough to make the temperature nice, but it's still cold. If it's an automatic control system, the difference between where you want to be and where you are is still what it was before. You apply another control action. And of course, you do that 5 times, yes? All right. Then the control action you took the first time now takes effect. And you say, now this is good. And then you don't do anything, right? You haven't done anything. But the next thing, temperature gets a little warmer than you want. And then maybe just a little too warm. And now you start cranking cold water. But the more cold water you put, what happens? The hotter it becomes, right? And just drives the automatic control crazy. And what do you get? You find these oscillations, yeah? Well, the same thing happens in biological systems. If there is too much of a distance that the molecule has to travel, and if you're just saying, well, I put in the hormone, I haven't seen any difference, well, why don't you just put in some more hormones? Well, you don't want to do that, okay? So these are the kinds of characteristics that we see in actual engineered systems. They are part of the biological system. So anyway, feedback control is, we understand negative feedback control, but there's also positive feedback control. Now, the minute I say positive feedback control, everybody is thinking about, if I stand in front of the speaker, the loudspeaker, with this microphone, what happens? You get positive feedback. It builds up, and then you have this ear-shattering thing, right? And you want to move away from it, right? Well, there's actually a part of biological systems that uses positive feedback control. When you want to build things up very quickly, when a woman goes into labor, oxytocin, you want to speed it up so that what needs to happen can happen very quickly. The same thing happens, it's very rare, but the same thing happens with platelet aggregation and blood clotting. Because if you cut yourself and you start bleeding, you better do something or you can bleed to death. So you want to very, very rapidly get the platelets coming to the site. And what happens is the platelets are moving right along. They run across collagen. I'm going to talk to you about that example a little bit later on. They run across collagen. They bind because the platelets have receptors for collagen. They bind, and the minute they bind, they degranulate, and they send out signals. Any platelets out there, we need you to come over here. There's a party going on. And they all get the message, and they come. And as soon as they come, they bind and they degranulate. They call others too. You can see how this is going to get out of hand very quickly. Right? If I'm here and I call for 20 people and they come and 20 people call for 20 more people, and very, very quickly you build it up and you plug the hole. That's why you use positive feedback control in many cases. All right? So feed-forward regulation, I'm not going to talk too much about that, but if you go outside when it's very cold and you don't have a jacket on, you start to shiver. That's feed-forward control. What you're trying to control is your core temperature. Your core temperature hasn't changed yet. Okay? Feedback control, the variable you're trying to control has to change first before you respond. Your core temperature hasn't changed yet, and yet your brain is telling you, you need to do something because there's a disturbance right now. You start to shift thermoregulation, and a whole bunch of things start to happen. Very close to noon today, if you walk by a restaurant, you're hungry, and you smell food, you know what happens? Your mouth starts to water. That's feed-forward control. Your system is getting ready for eating food and things of that nature. Cascade control is really the way the endocrine system handles the problem of time delays. Okay? If I'm going to send a molecule to go and work at a distance, that's the way that works, because it's a chemical molecule that works at a distance. In fact, the design of the endocrine system is a fantastic thing, because you're using the same highway that is general to achieve specificity. Think about that for a second. All of these molecules are traveling inside of your blood vessels and things like that, and yet they have very specific things they need to do. They have to bind to specific receptors for them to act. So two things happen. First, you have cognate shapes. So you have a chemical molecule that probably has this kind of a shape, a protein that has a shape like this, and then you have a receptor that has a shape like this. And it would only bind when the shapes fit. So if I'm coming and I have a square shape, and I'm coming and I find something like this, I just pass right by because I can't bind. All right, but they have to act at a distance. So they're going through this distance. It's going to take a long time for them to get there. So if I'm generating the molecules here, and I'm going to act over there, that's going to be tough. So what does nature do? Nature evolved such that what you have is a primary controller, and then a secondary controller, and then a tertiary one, and then you have this cascade control. So you act on this, and then you have a feedback, and then you have act on this, and then you act on this, and then you act on this. So that you will see a whole sequence of hormones that fit into this same mode. That's why you have cascade control in these kinds of systems. All right. So characteristics of your biological control system, I'm just going to go through this very quickly. It's hierarchical by definition. It has to be. Because you have the whole organism, then you have the organs, and you have the subsystems, you have the molecules, you have all kinds of things. So it is hierarchical. You control at different levels. It is massively parallel. Massively parallel. Just think about your immune system. I don't think I have a diagram to show you about your immune system. You have cells all over the place that are, you have the B cells, you have the T cells. I think I'll show you one of them. It's massively parallel, and yet they're coordinated. It's redundant. It has to be. So that if something goes down, something else can take over for it, and so on and so forth. So I'm going to skip all of this. And naturally, it's multivariable because there are multiple variables that you're trying to control all at once. It is highly nonlinear, which is actually an advantage, as you will see a little bit later on, because then you can get very fast response when you need it, and you can slow the response down when you get to where you need to go. If everything was linear, then you would just keep responding that way, and you would overshoot and undershoot and things like that. So it's very helpful. It's adaptive. It has to be. And generally, globally asymptotically stable. I mean, they're mathematical models. Yeah, they're models, but they capture enough that shows you how this concept of homeostasis comes about, because any perturbation just naturally wants to come back down to where it's supposed to be. I'm going to skip through all of this and get to, this is a cartoon, a simplified cartoon of the human immune system as a control system. So what happens, you have this antigen population. There are all kinds of things in this room right now, okay? And we're breathing and we're fine. So what happens is you have this sensor system, which is broken into two. You have your antigen processing and presentation. They make the antigen easier to recognize. They make the antigen, it's possible for you to tell not just that it's an antigen, but what kind. And so this is what you do here. And then your T cells, specific receptors, and the B cell antigen specific receptors can now pick up on these things and then go through the B cell effector system and the helper T cells and go through a whole, these are your actuators and so on and so forth. Without going into a lot of details, do you know what the AIDS does? The acquired immune deficiency system, HIV, takes that out. So now you can see if you take out your primary controller, you're susceptible to just about everything. And that's why it is a very, very dangerous thing. All right, so let me get to the next phase of what I'd like to talk about. And pathologies, control engineering perspective. And two things I'd like to talk about. If the involvement of feedback control in a biological system is not recognized, if you just naively look at input-output data analysis, you will get erroneous results. Let me give you an example before I go. imagine that as we're sitting in this room, that it's very hot outside, just for the sake of argument. All right? And so we keep opening this room and closing it, and opening this room and closing it, and opening and closing. If the control system, temperature control system in this room is very good, what do you think will happen to the temperature in this room? It will not change. Because it's responding to everything that's coming. It responds very quickly, so you don't see it, you don't know. And so if you then try to correlate the outside data with the inside data, you will wrongly assume that any variation in outside has no effect on the inside temperature. The reason is you have feedback control that is taking that variation, transferring it to variation into something that you haven't looked at. Okay? So if that is the case, be very careful how you make your conclusion. I'm going to show you an example of a case of TGF-beta in cancer, okay, where physicians and the researchers in that area almost made a mistake because they did not recognize the fact that there was feedback control involved. A control engineering perspective of diseases mitigates the susceptibility of fault diagnosis. Let me ask you this. As engineers, as control engineers, if all of a sudden this room starts getting too hot and you know right away that the temperature control system is not working, I ask you, what is wrong? Would you know? Well, it could be the sensor, right? The sensor may not be working, everything else is working, but the sensor is not working. But it could be the controller. That's the controller that's not working, the sensor is working. Or it could be the HVAC system that is not working. In fact, it could be this room itself that's not working because somebody punched a hole and it's allowing air to leak into this room, okay? However, if you are looking at somebody who shows up and says, I have, well, you take the measurement from the blood and you find that the calcium is very, very low. Well, you have hypocalcemia. Okay. But what kind? What's wrong? Why do you have hypocalcemia? Okay? That's a challenge because it could be a sensor that's failing. It could be a controller that's failing. It could be an actuator that's failing. For that matter, it could be your system itself that's failing. So we need to know which is which, and yet it is not that easy to do that diagnosis. It is not medically. But if you at least have those concepts in front of you, then you can start thinking, where can I perturb the system so that I might have a better chance of being able to isolate what is wrong? So this is what I'm about to show you. So I've just explained to you the difference between open loop and closed loop response. If we're talking about open loop response, I changed the temperature outside. Without feedback, the temperature inside will change. But with feedback, it may not change. All right? So many diseases arise from a control system component failure. Stop and think about that for a second. Everybody understands diabetes. Okay? Diabetes means that your blood glucose is not being controlled well. However, it is possible that it's not being controlled well because you simply don't make insulin. Okay? You don't. And if you don't make insulin, that's the problem with your controller. Your measurement of blood glucose is fine. It is getting the information to the pancreas, and the pancreas is saying, sorry, can't help you here. But in another case, it is possible that all of those things are fine. You're actually making insulin, but for whatever reason, your system is no longer responding to insulin. The gain has gone close to zero. Right? That's type 2 diabetes as opposed to type 1 diabetes. These are control system failures. These are understood in the medical field and they are treated accordingly. But there are others that are not understood that way. So many diseases arise from control system component failures. Appropriate diagnosis and treatment requires the correct identification of what has gone wrong. If you use traditional input and output data analysis, you are not going to get the results because if you just do input-output, you conflate everything together. You have to be very careful. So I'm going to zip through these very quickly. I'm going to talk a little bit about calcium regulation. I started giving you an example. And then the TGF-beta in prostate cancer, confusing the role of TGF-beta in late-stage cancer. And this is the problem of confusing correlation with causality. So let's talk about calcium regulation. I worked with a group of people who they've spent all of their lives working on calcium regulation. One of them is a nephrologist, and it's difficult for me to explain why somebody who deals with kidneys is working with calcium, but it is. There are a lot of connections. But anyway, the idea was for us to understand from a mathematical and quantitative way how calcium is regulated, and then employ the model for diagnosis. A few quick things. 99% of the body's calcium is stored in the bone mineral. I'll show you a diagram very soon. And the other 1% is distributed between the ECF, extracellular fluid. And so, give or take, this is pretty much the range that you're looking for. For those of you who are chemical process control people, this is a rather fairly accurate process representation of how calcium is maintained in the human body. Think about the bone as a reservoir, okay? That's where you keep all of the extra. And when you need calcium, you take some of it out. And when you have too much calcium, you put it back in the bone, okay? So that's like a reservoir. And then you have this, see your heart? It's pumping these constantly through. You're absorbing calcium from the food that you eat. You take food in. That's like an extruder, and you extrude out the waste. And as you're doing this, you're taking, this is your osteoblastic activity is taking calcium from the blood, and osteoclastic activity is taking it from it and putting it back into the bloodstream. Your kidney, this is why the nephrologist works with us on this, because your kidney plays a central role in recovering calcium from urine, and you lose calcium to. And anyway, there's a lot going on, and I really enjoy this, and I wish I had time I could get into the physiology of how all of this works. But last but not least, I'm going to talk about the parathyroid gland. The parathyroid gland is the controller, and it also has the sensor. So there it is. It senses what's going on, and then it starts to orchestrate what's happening. All right. So this is a fairly accurate diagram. And so what happens is if you first sense, so for example, that maybe your calcium is low, the first thing that happens then is your parathyroid gland sends a message, and anything that you see that's solid, any solid thing is the release of a material. Anything that is dashed is information, okay? So it releases parathyroid hormone, which is going to do something to the kidney. It's going to stimulate the kidney to produce calcitriol. And in producing calcitriol, that does several things to the gut and to the bone, and a whole sequence of events start to happen. And without going into a lot of details, how many of you have been around a pregnant woman who all of a sudden wants to eat ice cream and sauerkraut? The reason that happens is because you're at a stage where the bones are starting to develop in the fetus, and you need all the calcium that you can get. And so while you're pulling the calcium from all over the place, and you say, I don't have enough calcium, that's the calcitriol talking. It says, go get me some calcium wherever you can. And that is why the wife would scream at the husband and say, go get me some sauerkraut and some ice cream, will you? Okay, but that's part of what's going on here. And if you put the control block diagram together, I'm not going to drag you through all of this. This is what you get. This is not your standard control system, but you have your parathyroid hormone acting on the kidney, and so you have your controller here, and you have the kidney acts as when it's producing calcitriol, it's a certain kind of an actuator, but when it is reabsorbing, it's a different kind of actuator. But anyway, you put all of these things together, and you fill this with mathematical models. And in filling them with mathematical models, we can now look at what's going on. We just published this paper in the Journal of Clinical Endocrinology and Metabolism. All right, so ladies and gentlemen, if you really want some drama as an engineer, try publishing a paper in the Journal of Clinical Endocrinology and Metabolism. Okay, there was quite a bit of drama. Well, in part because we speak different languages. I mean, completely different languages. But with the help of our physician friends and things like that, we were able to make sure that what we're trying to say is understandable and things of that nature. So, cut a very long story short, you put a model like that together, you need to validate it. And so we took some clinical data and we validated the response to induced hypocalcemia. You induce hypocalcemia with a pulse, and the red dot is the data, and this is a mathematical model. But what our model can do is that it can, in fact, this is validation with hypercalcemia. So what we did here was to fit the parameters that we didn't know to data for hypocalcemia. And then we validated it with data for hypercalcemia. So in one case, you reduce the calcium. In the other case, you increase the calcium. It's a nonlinear model, and if you can match it going down and up, then you've really done well, and which is what we did here. All right? So, but what our model can do is that it can tell us what's going on intra-measurements. Okay? After you stopped what's going on, for example, the calcium in the urine would behave like this, although it wasn't measured. Okay? It was not measured, but we can predict it because we have it as part of our model. The parathyroid hormone, which was not measured, will behave like this, and calcitriol will behave like this. And what's going on with with the ratio of osteoclastic and osteoblastic activity. We can predict all of those things, which is going to be very helpful for what I'm about to tell you. When somebody has hypercalcemia, it is possible that you have different ways of developing hypercalcemia. So primary hyperparathyroidism, called PHPT, is responsible for about 70% of all the cases. Okay? But you also have this familial benign hypercalcemia, FBH, or humoral hypercalcemia of malignancy. So somebody shows up and you have hypercalcemia. Is it this, or is it this, or is it this? If you just want to guess, which is what most people do in this case, is if you come and it's hypercalcemia, 70% of the time is PHPT. And so we'll treat you for PHPT. But you may be part of that 30% that is something else. Well, then you have to do a few other things to be able to figure out if it's this or this. And it's not necessarily easy. All are classified as hypercalcemia. Similar presentation, you see high calcium and high PTH-like effects. Okay? Differential diagnosis is very difficult. So I don't know how much time I have left. Not that much time. So the root cause of hyperparathyroidism is the abnormal enlargement of the parathyroid glands, and following diabetes and thyroid disorders is the third most common endocrine disorder. And so here are the characteristics. You see chronic hypercalcemia, sustained bone loss, and then 10 minutes. Okay, all right. So with 10 minutes, I'm going to have to run through all of this very quickly. The problem in this case is a controller defect, because it is your controller that is not working. The familial benign hypercalcemia, in this particular case, it's a missense mutation of the calcium sensing receptors. And it's familial because there's a genetic problem involved. This is a sensor defect. Okay? It's a sensor problem. And then the humoral hypercalcemia of malignancy, the problem is if you have certain cancers, breast cancer, lung cancer, and some blood cancers, the tumor cells produce what is actually called PTH-related proteins. It behaves like PTH, but it's not PTH. PTH receptors detect it, and then they act as if it's PTH. And so now you're putting PTH out as if you have a problem with calcium, but you don't. It's because of something else. And then that causes bone loss, reduced calcium excretion, and a whole bunch of things like that. Anyway, this is an actuator disturbance. And so in the end, it turns out that in this particular case, you can break them down to different things, and the way to then use a mathematical model, and this paper is currently in press, So we're going to validate our control system. We're looking at this from a control system perspective. We're going to validate that we can use this. In other words, can I reproduce the clinical observations by making changes in my control model? If I can do that, then I'm going to simulate a few things to see if the things that are not currently measured can be measured to help with the identification. And that's what we did, and I'm just going to run through this very quickly. This is classic. People who deal with calcium in the clinical area, this is what they use. It is an input design in which you, at time 0, you put in 1 milligram of calcium, depending on your weight, and then at 1 hour they increase it to 2, and then 2 hours they take it out, and then they watch what you do. They watch your response. So this is data. This is our mathematical model of all of these things. This is the response relative to baseline, and it's difficult to see. Okay, this is for FBH. This is data for FBH. This is our model prediction. And then for, so let me just show you real quickly because I'm starting to run out of time. If you look at this very carefully, this and this is the response of your PTH relative to baseline when you're healthy and when you have FBH. This is because it's a sensor problem, all right? So the normal behavior and the sensor problem behavior would look similar, all right? And so if I were to test you and I know you have hypercalcemia and your response looks like it's normal, but I know you're not normal, then chances are you're FBH, okay? Because of this characteristic. Look at this red line. This is PHPT. This is completely different from this, all right? So when I induce that and I see this kind of response, chances are you have PHPT. All right, and there are a few other things that I could show you, but in the interest of time, I'm going to move right along. The TGF-beta problem is that clinical data showed that the people who have the worst prognosis of prostate cancer had the highest level of TGF-beta in the tissue that was taken through biopsy. Now, that may not sound like a crazy thing, but TGF-beta is known as a tumor suppressor. So let's stop and think about this for a second. How come a tumor suppressor, as it's increasing, your prognosis for prostate cancer is getting worse? Makes no sense. It is almost like somebody saying cooling water is going to keep going up as your temperature is going up. Or the temperature in this room is going to keep going up as we keep cranking cold air into this room. It makes no sense whatsoever. Well, what if there's a controller involved? And that was the case. That in this case, so we have a control engineering approach to understanding TGF-beta. This paper is out in the Journal of Royal Society Interface. And we put it together as a control system, and I don't really have time to go through all of those, but it was a very interesting thing to see how the actuation is done with TGF-beta actuation. But anyway, be that as it may, if you put it together and you look at the response to a step change in the cell population, this is how your system behaves under normal circumstances. And yet, when you start simulating what happens with prostate cancer cells, the problem is prostate cancer cells lose their sensitivity to TGF-beta. And therefore, in a futile attempt to bring the cell population down, the controller keeps cranking in more TGF-beta, and it keeps cranking in more TGF-beta, and there is, in fact, there's a mathematical model that you can use to do some stability analysis that shows you why then you will cross a certain point where the amount of TGF-beta is going to keep going up as the cell population goes up. And the problem, of course, is that you did not recognize that this was the case. So I'm going to skip through all of this and then finish. This is the stability analysis I was talking about. And let me very quickly just talk about this last thing in 5 minutes. Talk about engineering control systems and disease treatment. In this case, I'm going to talk about platelet count control. What I'm about to show you is an ongoing work with a real patient, a real-life patient. And I'm going to tell you real quickly what's going on. The point I'm trying to get across here is that when the biological control system fails, as it does every now and then, with proper recognition and paying due respect to the biology. You need to do that. It is possible for engineered systems to help. Okay? And you know some of the work that Frank Doyle does with diabetes and things like that. This is another area. All right? So, hemostasis, not to be confused with homeostasis. Okay? Hemo is blood. Hemostasis, this is the Greek has infused a lot of medicine. Hemostasis is the system that stops bleeding when you cut yourself. And I started giving you an example of what happens. You have two things. You have platelet aggregation, which brings all the platelets in, but platelets just form a plug. It's not enough to plug it up completely. Then you have what is called coagulation, where you have the fibrinogen mat that makes it strong. Okay? So you put the two of them together, you can then plug it up. Unfortunately, ladies and gentlemen, what happens if you don't have enough platelets? That's what you use to plug it. If you don't have enough, you can't plug the hole and people can bleed to death. Okay? The flip side of that is, what if you have a system that is so sensitive that the slightest nudge, your blood clots? Then you can have all kinds of things. You can have a stroke, you can have deep vein thrombosis, and all kinds of things like that. So you need to maintain a particularly good control on this, and I'm not going to go through all of this, but this is the block diagram. Imagine then if somebody takes out your platelets, okay? And this is what happens with ITP, immune thrombocytopenic purpura, all right? And you need your platelet count to be somewhere between 150 and 400, that's times 10 to the 9 cells per liter. And when you get low to 30, you have a high risk of bleeding. Less than 10, things are very, very serious. So in this particular case, let me just skip through this. How do we make platelets? We make platelets because of TPO synthesized in the liver, circulates through the blood, binds to the precursors, you make platelets. Now, classic feedback control in biological systems. Platelets themselves have TPO receptors. So if you hang with me for a second, TPO is what helps us make platelets. But platelets have TPO receptors. So the more platelets you make, they take some of the TPO out so that you don't overmake platelets. Okay? So this is they come back and inhibit this, and it lowers the concentration. So that's the typical thing. How is ITP treated? It is treated with this disease, with this drug called romiplostim. Romiplostim reacts, behaves like TPO. We don't know why, but it does. Okay? It behaves like TPO. So you can use it to make this, but it takes days. The challenge is very expensive, and you have a long delay before the effect can be observed. Keep that at the back of your mind, because delays and feedback control can cause all kinds of interesting things. All right, so the objective of this study that I'm going to try and finish very quickly is for a particular patient, they just couldn't, for years, maintain this platelet count at 70. It is always very high, dangerously high. If your platelet count is too high, you can get plugged up very quickly. If your platelet count is too low, you can get yourself into trouble and bleed to death. Okay? Treatment limited to pulses of injections, fixed magnitude at discrete points in time. Current practice is limited to weekly or biweekly treatments. Okay? So we developed a mathematical model. We analyzed the model for insight. We used it to develop and evaluate control schemes. And these are the three control schemes that we looked at. Fixed dose, open loop control, which is the current practice. Okay? Fixed dose. If you show up, they take your count and then they give you an injection. Okay? And then they come back 2 weeks later. Optimally tuned PI control and variable dose optimal open loop PI control. Let me just go through this very quickly. This is real data for the patient. These are the treatment spikes. We fit our mathematical model up to this point, and then without changing anything, we predicted what will happen later on in the future. Now you'll see a few things that they don't quite match. That's because of a lot of other things, but it captures a lot of what's going on. What I would like to point out to you, though, is this characteristic of the model. This T platelet is the lifetime of your platelet. For a normal healthy patient, it's 253 hours. For this particular patient, it's less than half. So the platelets that you actually make for this person dies off in half the time. That's part of the problem. Anyway, here are the characteristics, the open loop response of your romiplastin. If I give you, at this initial platelet count, close to 0, and I give a 1 microgram per kilogram, this is the response you will get. And as I increase the number of platelets, initial platelets, notice that you get less and less of a response because of this feedback. By the time you get to 250, you get nothing. You get no response because you have enough platelets. You don't need more. You give me the romiplastin, I don't respond to it. This is the flip side. From the same platelet count, and if I give you different doses, you get this kind of response. And you can see it's nonlinear, and they get closer and closer. All right, so very quickly, let me show you some of the results. If fixed dose open loop, 2 micrograms, this is weekly, this is what the injection, this is the response you get. Which, if you remember the data that I showed you earlier for the patient, this is what they were seeing. And the doctors were really concerned. They were never able to maintain it steady. And of course, this is what we're predicting too, that this is what you're going to see. If you do it every week, bi-weekly, you get this kind of response. If you do 1, you see this response. If you do half, in neither case are you able to get it to be steady. All right? However, if you tune your PI controller optimally, it is actually possible to get this kind of a behavior. This is once every week, ladies and gentlemen. If you do it bi-weekly, you get these measurements that give you the illusion that you're doing well. But look at what's going on in between. Okay? And this is what gave us the hint as to what was going on. And I'm going to skip this. Here's the problem. The doctor was trying to find out, why is it that regardless of what we've done for years, we've never been able to stabilize this patient? Here's the reason. This is the open loop response of an injection of romiplastin. If from when you inject to the peak is approximately 2 weeks. Ladies and gentlemen, this is the natural frequency of this behavior. So if I show up and I get treated every 2 weeks, what do you expect? The response you're going to find is a nonlinear convolution of the basis function. I mean, that's essentially what it is. If this is the basis function and you have a nonlinear convolution of this is what you're going to be seeing. So the recommendation is we really need to change this. Not treat every 2 weeks, start treating every week, and then maybe we need to start using a lookup table in recognition of the delays and things like that. It's really simple enough to do, and we're actually in the process of exploring some clinical trials. All right, so let me conclude very quickly. The central issue is that control is central, an inevitable feature of biological systems. Without control, none of us will be able to. I'm walking, I've been talking and doing all of that, and I haven't killed over. Okay? Without control, I would have killed over completely. All right? So a control engineering perspective is advantageous. It helps us with efficient modeling. We get analytical tractability, and if all we did was just collect data and more data and more data, it becomes very difficult to see what's going on. Effective diagnosis. Medical schools love recruiting engineers. I'm starting to find that out. Many of our students are leaving a bachelor's in engineering and going to medical school because medicine today has become very complex, and people are recognizing that it's a combination of all kinds of systems, and engineers naturally trained to think that way, but you need to go learn the medicine, you need to learn the biology, and then put all of these things together. So we've started incorporating principles and methods of biological control systems into our graduate curriculum of teaching our students. Here is a collection of all the people that have worked with me over the years. Chair Hong Tai is a current grad student. This is a postdoc, and all of these guys have graduated. And so all of my collaborators at Thomas Jefferson University Medical School, these are all, now he's now at TJU. Anyway, that's my story. These are the funding sources, and here's a group, research group photograph from 2012, and I'll be happy to answer any questions that you may have. Thank you for your attention.
Well, this was a wonderful talk, and I guess engineers have a lot to learn from biological systems, and doctors have a lot to learn about establishing the conventions of biological systems into medical problems. And I think that the only barrier that we have is actually what you said, language.
Yes.
And I guess people in faculty of medicine have this phobia of anything that's quantitative and has equations and vice versa. Yeah. So, well, anyway, Professor will answer some questions, and what we'll do is that until 11 o'clock, then at 11 o'clock, people who want to leave can leave, and the interested can stay for a little while, because there's nothing else in this room. So, any questions?
Yeah, I have a question about death.
Mm-hmm.
There's no... is it working? Yeah.
Yes.
There's no living organism, no control system that has worked forever. I mean, multicellular, unicellular.
There's no control system that has...
No biological control system that has worked forever.
Ah.
They all end up stop to work.
Yes.
Even unicellular organisms.
Yes.
So, is it ever going to be possible to stop that?
That is a heavy philosophical question. And I would answer it two ways. First of all, there's also no humanly engineered system that works forever, right? It would die at some point and you have to replace it. So that's just, anybody who studied thermodynamics would understand that's the natural thermodynamics of everything. However, you raise a very, very interesting philosophical and ethical question that many people in the medical field are starting to think about. Imagine that your liver fails. Normally you would have died, but now we have all kinds of things that can be, artificial liver that can do some perfusion and things of that nature, or you do dialysis for your kidney, or this or that. People have actually started to speculate that not too long from now, you will have the mixture of cybernetics and physiology, and by the time you know it, who's a human being anymore? When your eye has been replaced with some electrical thing that is actually better than what you had there before, and your kidney has been replaced with something. Right now, knees are being replaced. People who've lost their limbs have received things that actually function better. And so you start doing this, and then a human being is 75% organic, 25% this. And by the time you get to 95% whatever and only 5% organic, people could live forever. I mean, I would hate to live in a world like that, quite frankly. But you raise a very, very important ethical question. Maybe you want to answer some of that from the medical perspective.
What about the death of unicellular organisms? Mm-hmm. The problem starts at the one-cell level, right?
Yeah.
Have a question here?
My question is about stress, which is so common nowadays and causes so many problems to people.
Yes.
Well, the question is it an open loop? Because it seems to me that it's completely open loop. And if there is any research about trying to detect and to control stress, would be great.
Yeah. Actually, it is not open loop because there are stress hormones that are detected in the body, and the body responds. In fact, what you call stress, the stress response, is a closed-loop system. Imagine if a bear comes into this room, or if I shout fire in this room. Please don't run away. If I do that, then immediately it's parasympathetic and sympathetic systems. There's the flight, right? Your adrenaline and all of that is geared towards making you escape. That's what you call stress. Sometimes is a feedback response to things that are happening to you externally. And I'm forgetting the name of the stress hormone now. Well, adrenaline is one of them. There's another one, ends with OL. All of them ends with OL. But anyway, it's when they build up cortisol. Thank you. It's cortisol. It's when they build up too much in your system that starts to destroy your cells and things like that, which is why you really shouldn't do those kinds of things. Which, by the way, there's another aspect of all of this that is the intersection of medicine with psychology. Why is it that the Buddhist monk that meditates doesn't have as much stress? Okay? What does meditation do? People are starting to study that, because when you meditate and you breathe in and you do all of those things, it actually dials down. People have, there's clinical evidence. It dials down all those responses. What are the tracts for those kinds of things happening? We don't know. But you're right that stress is an external disturbance. And then we respond to that external disturbance, and some people do it better than others, which is why if you don't get up in the morning or in the evening and go run and exercise, because your system then releases all kinds of things that counteract all of these other things.
Two-part question, I'll keep it
Yes.
short. Do you see the need for new control theory? Most of the structures you described were fairly conventional, but I'm sure you've seen some things. You did mention very quickly. And the other part is, what's the return to engineering? I already asked you this 6 years ago, but
Yes.
maybe there's a new answer.
Well, the first part of this is some of the things I've shown you are cartoonish. Biological systems are notoriously complex. They're just notoriously complex. And so the questions that we need to ask ourselves is is there some theory as to why this particular steady state is globally asymptotically stable, and then when something happens, as in cancer, it completely takes out that attractor. Because, I mean, that's really what happens. Otherwise, you get back to the attractor. That's why it's called an attractor. So what are those kinds of things? And so there's room for understanding the fundamentals of why things like this happen and why they fail. Now, returning to engineering, in what sense? In having engineering come back to help biology, or?
No, getting ideas from biology back into designing better control systems. Biologically inspired.
Actually, yes. I wasn't able to publish any of that, but at DuPont, we actually did some of that work, because that's where all of this work started. And I can tell you this one because we published it. The blood pressure control system, the sensor system, is uniquely, it's breathtaking in the way it is put together. You have C fibers and A fibers, some with myelin sheath around them, some without. Some encode dP/dt, the temperature change, some pressure change, some encode P by itself, some very noisy but very fast. And so you have all of this nonsense going on, and yet somewhere in the NTS it is integrated together, and you get one of the most robust measurements you would ever face. And in fact, my collaborator deliberately did things like cut some of these neurons and just take them out and see if it would affect. No effect whatsoever, none. And so how do you do these kinds of things? All right, so then we had a particular problem. We have a polyethylene process in which the reaction, I'm not making this up, the reaction is over in 12 seconds, okay? It's like running 100 meters. 12 seconds, it's over. But it's open loop unstable, and you get out of range by 2 degrees, you can blow up the whole place. So the only way you control what's going on is by control the quality, is by knowing what you put in. Okay, so the ethylene and the copolymers, and so we have several measurements of the feed. Reaction is over in 12 seconds, okay? So you better know what you're doing. So we have measurements from gas chromatographs, we have measurements from all kinds of things, and then we have a thermodynamic model to tell us what's going on. And then we take samples, we take it to the lab, And then after 30 minutes, we find out what's going on. Reaction is over in 12 seconds. So they came and asked us, which of these measurements should we use? And we said, wrong question. Use all of them. Because that's what biology does. The trick then is figuring out how to do all of it. Well, we published this paper, so I'll be able to send that to you. The other advantage that this helps us do is the nonlinear characteristics. I don't know if you saw, but the baroreceptor reflex, yes. And so instead of having a gain that is just Kc, if you have a sigmoidal gain, you get some of that advantage. And there's several other things that you can be learning. When all of us were born, our neonates, the neonates don't have anything to control standing up. You can't. And then you start learning how to walk. Well, there's a very interesting way, and people have followed how neonates learn how to walk and how to stabilize themselves. By the way, standing up, we are bipeds, we're open loop unstable. We have a pair of complex conjugate poles, one in the right half plane, the other one in the left half plane. Okay, so you have to stabilize it. And so that stabilization is learned through iterative learning. Kids get up, they stagger, they fall, they get back up again, and you learn. We can also benefit from that sort of thing. So,
the way you presented, you decouple these systems to analyze and to design the controller. But in some point, you have to couple again. So how decoupled and how coupled the system could be treated?
They are coupled, and the thing that we all need to understand is with biological systems, you are better off taking subsystems and then putting them back together again. Okay? So otherwise, if you try to take them all at once, you're really not going to be able to do anything well. So the way we put it back together again, and it's like a chemical flow sheet. If I have a chemical process flow sheet, I have my mixer, I have my reactor, I have my distillation column, I have this, I have that, and I can study each one of them, and then I can put them all back together again, which is what we did. We studied individual systems, and then you put them back together again. If you're talking about the entire organism itself, some people have tried to do that, but I'm not one of them, because I would rather focus on certain aspects and treat anything else that's coming from those things as disturbances. Okay? You lose something because it goes both ways, but especially if they are physiologically removed, then it's not going to be that much of a disadvantage. If you saw my calcium homeostasis diagram that I showed you, I had my circulatory system, I had my digestive system, all of them interacting. And to put all of this together, I included those that had direct influence. What I did not include were things with the nervous system, although it has something to do with it. Okay? But we can discuss that a little bit later on. That's a very, very important point in being able to get a handle around biological systems. They are notoriously complex. And if you try to do it all at once, you would not succeed. So you take it off bit by bit. It's the old story of how do you eat an elephant, one little bit at a time.
Well, Professor,
yes.
Well, my question is related to the last one. It's quite related to Antonio's question, because you put human being systems or biological systems as a natural hierarchical control system, and this is true. And so when you talk about treating a disease, so what you did was you isolated a subsystem, and you said that it was important to design the control law to identify where the problem was, if it was in the sensor, actuator, or control, or even in the plant system. My question is, once you detected where the problem was, okay, so you could design a control law for the system knowing that, but you could also think that maybe the problem now could be seen as a lower level where your control system to be treated is the component that where you identified the problem. So you would refine your model, and instead of changing the control law to solve the problem, you would replace the control problem as a problem in this component. And so I would like to know how you think about this.
Yeah, if I understand you correctly, it is maybe the difference between a control problem and maybe even a design problem, or the fact that things do interact. And therefore, by focusing on one level and identifying what's going on, whatever it is you do may in fact influence other people. You have a very valid point, and I think just like we would, if we're dealing with a distillation column in a refinery, and we notice that the problem is with the distillation column, I'll deal with that first. And if something else shows up, then I'll deal with that one too, rather than trying to take everything all together. And in terms of being hierarchical, that's actually an advantage, because then you can identify at what level you've seen a problem and make sure that it's not any higher or any lower, and then deal with it at that level. And if something else happens, then you might want to pull yourself back and take a look at it. But if I didn't answer your question correctly, let's meet afterwards so that we can finish the answer.
Any further questions?
I might have one.
Okay.
So, Professor, you were actually showed us all the responses for the control systems that you came up with. But when it comes to the question that the lady made, when it comes to aging, do you take into consideration that factor as well, or the curves response are pretty much discrete, working or not working?
That's a very good question. If you're working with a patient, the concept of aging, as you probably know, is one of those long responses. So it's almost like catalyst decay in a catalytic converter. It doesn't happen over a week or 2 weeks or things like that. So it takes a while for it to evolve itself. The particular patient that we're talking about, a few years back, if we had used that data for this model, the platelet life would have been slightly different, actually. And so as long as you're dealing with real systems, these are quantifiable parameters. Aging is really nothing but you think about it like your actuator. When you're walking, the actuator gain is changing. So you can identify these things. So with time, you can track them. The good news is it wouldn't happen between 2 visits to the doctor. It will happen over an extended period of time. So it allows us enough time to track if we need to track. And yes, it is eventually, I'm sorry, once we cross 40, it's eventually going to go down at some point, and that gain goes to 0 and we all expire.
Well, there are no further questions.
Oh.
It seems there are some organisms that find a way to solve that. Lobsters, it seems that they live forever, at least 54 years. Difficult for them is to change the shell. So can we learn about that and use that in our models?
Well, you raise another important point. One of my former students actually looks at regeneration. If you take a worm and you cut a worm, right, it grows back. And in fact, the liver, if you take a liver and you cut off a certain portion of the liver, it not just regenerates back to exactly what it was before. Exactly. If you haven't drunk too much alcohol, if you've drunk too much alcohol, no, seriously, it affects it. So there are issues of regeneration that people are starting to look at, not just in higher order mammals, they're looking at lower level things, and there's certain things that allow people to do what they do. You geneticists tell us about telomeres. You take something, you replicate, it's like a Xerox machine. You're allowed to Xerox only so many things, and so you Xerox that cell so many times, that's it. You can't do it again. Well, there's some things that they're finding out that you can do tricks with the telomeres. So I've told you a little bit of what we know. We're just scratching the surface. This is what makes all of this so fascinating, because there's so much more that we don't know, and what's going to happen in the future. I think with engineers working with the people in the medical field, we might be able to move faster if we can figure out a way to understand each other's language.
Yeah, I guess that you can fool the second law of thermodynamics for a little while by influx of energy and keep it away from equilibrium, but in the end, you're going to go back to equilibrium.
It catches up to you. Yeah, it catches up to you. Somebody described the laws of thermodynamics this way: you can't win, you can't break even, you can't get out of the game. So it will get you eventually.
I'm just curious about something, because I was an ITP patient myself as a kid.
Ah.
Yeah, I had when I was a child, and I got well.
Yes.
But what I remember is that they didn't have an explanation for what caused the disease.
Yes.
And as I saw, you look at these diseases as control or sensor failures in a control system. But what I would like to ask you is that if that helps, like, to shed a light on what causes this kind of failure in different sorts of diseases.
I'm sorry to tell you, the I in ITP sometimes is called idiopathic, which means I haven't the foggiest clue what caused it.
That's what I imagine.
No, fundamentally, we don't know what caused it, but at the next level, you know that you were not making enough platelets.
Or
Why? Destroying them at a higher rate, or both. And so children, actually, this is fascinating, and if you don't mind, I wouldn't ever use your name, but I would tell people the next time that children actually can overcome ITP.
Sure.
And so...
Yeah. Most of the times it's a short-lived disease.
Yes.
Sometimes
it is, yeah.
That's another problem for control, right? Why some are short-lived and others are not?
Well, people actually suspect that with children, there were stem cells and things like that. Whatever it was got corrected with time, and genetics. I mean, you're the expert in this area. And genetically, things get corrected and you just don't improve. It happens more with children. If you get it late in life, it's much more difficult. Fascinating.
Well, I want to thank you for a brilliant...
Obrigado.